fellow classmates, my name is Jacob Woods, and I would like to introduce you to my presentation on parasitic interactions of salmonids in the Pacific Northwest. Before we get started, I'd like to explain why I chose this topic. Salmon are of great importance in not only the vast majority of ecosystems within the Pacific Northwest, but also a great number of coastal environments worldwide. Their annual spawning runs from a marine to a freshwater environment serve as a primary source of nutrients for countless carnivores and omnivores within the immediate area. Salmon can be considered the biological foundation of many coastal ecosystems. The fishing and commercial industry is also extremely dependent on the success and preservation of these fish, which brings up the next reason I chose the subject. Fishing has always been something that my family and I have enjoyed over the past many years. These pictures were taken in Bristol Bay, Alaska 2 years ago when I spent the summer on a boat tendering salmon. Fishing is something I want to hand down to my children, and in order for this to happen, we must understand parasitic interactions that could harm salmon populations in the near future. So, to better understand some of these parasitic interactions, I chose to interview a professional. I was privileged to get to meet and interview with Charlene Hurst. Charlene recently received her master's degree in microbiology here at OSU after receiving her undergraduate from Cal State University, Long Beach. She is now currently working in a lab as a PhD candidate under Dr. Gary Bartholomew's mentorship. Most of Charlene's research has dealt with the parasite Ceratomyxa shasta and its interactions with salmonids of the Pacific Northwest. In the past 3 years, Charlene has been a part of 4 published articles dealing with Ceratomyxa shasta and its impacts on salmonids. Most recently, Charlene has been focusing on differential genotype susceptibility of C. shasta. Though most of her work deals with this particular parasite, Charlene explained that other parasites also plague salmonids in the Pacific Northwest. Lepiobathurus salmonis, or commonly known as the salmon mouse, is a relatively large type of ectoparasite. It is considered relatively large in that when fully matured adults are on a salmon species, they can be seen with the naked eye. These are ectoparasites in that once the free-living stage of the salmon mouse comes in contact with the salmonid, it adheres to the skin, where it then proceeds to grow from the nourishment provided by the mucus and blood of the salmon. This parasite has been of concern with many man-made fish farms and fish hatcheries. This is due to the close proximity of the fish, resulting in transmission of the parasite within the net cages. The infection of juvenile salmonids, the sea lice, can often kill the young fish. Another parasite that Charlene mentioned was Myxobolus cerebralis, also known as whirling disease. Rainbow trout appear to be very susceptible to this myxosporium parasite. The parasite requires 2 hosts to complete its life cycle, one of which is a tubifex worm and the other being a salmon. The infection of the parasite is very dramatic in juvenile fish, often resulting in death. The disease causes skeletal deformation, resulting in a corkscrew-like pattern of the fish's spine, making feeding and escaping predators very difficult. Fortunately, this disease is not transferred to humans. A parasite that has a very similar life cycle to M. cerebelis is Ceratomyxa shasta, also a myxosporium parasite that inhabits freshwater ecosystems of the Pacific Northwest. In order to complete its life cycle, it must undergo 2 separate morphologically distinct developing stages within 2 aquatic hosts. These hosts are a freshwater polychaete worm and often a juvenile salmon. One of the distinct features of the Myxosporium class of parasites is that they all require both an aquatic invertebrate and an aquatic vertebrate in order to fully complete their life cycle. This parasite is then separated into the genus Ceratomyxa for other distinguishable features. These features include the morphologically unique boomerang-like appearance of their myxospores, containing 2 polar capsules that often resemble eyes. Another distinction is that these groups often reside in the digestive tissue of their primary hosts. The most notable feature of C. shasta is parasites are unique in that they are only the freshwater-dependent parasites within the marine genre. This distinction may soon cause a change to the name of Ceratomyxa shasta to Ceratonova shasta, because this is the only species within the Ceratomyxa genus with a non-marine environment. After a salmon has already been infected with C. shasta and died, the parasite continues its life cycle by releasing myxospores into the freshwater environment. These small boomerang-like spores are able to thrive in warmer freshwater regions of the Pacific Northwest. The life cycle of C. shasta is progressed from this point once consumed by an annelid worm. Once this polychaete consumes the myxospore, infection to the worm may then occur. Research has indicated that the freshwater polychaete Maniukia speciosa is the most commonly infected invertebrate host. This worm is considered an intermediate host because the parasite continues to develop into a new stage, and also an obligate host because the parasite requires this specific host's internal environment in order for this transition to take place. The myxospore will then mature into an actinospore within the worm after around 49 days within the intermediate host. Once fully mature actinospores are present within the polychaete worm, they may then proceed to be released into the freshwater environment after being secreted through the worm's epidermis. Here they live in the freshwater until they come in contact with the gills of a salmon, where a route of infection may then occur. What is unique to Ceratomyxa shasta is that, unlike other myxosporan parasites, C. shasta develop into actinospores within the dermis and the striated muscle layer of the salmon, rather than within the intestinal epithelium like other myxosporians. Once C. shasta infects a polychaete, it may proceed to proliferate extremely fast, producing nearly 14,000 actinospores in just 2 weeks. These actinospores can remain infectious to the salmon for up to a week after being released from their immediate host. Ceratomyxosis, or otherwise known as enteronecrosis, is the disease that results from actinospores of C. shasta to salmonids. Ceratomyxosis, or enteronecrosis, is a disease that can affect all range of salmonids, with the highest prevalence seen in juvenile populations. These parasites can infect Chinook, or king salmon, coho, or silver salmon, chums, also known as gogs, pink, or humpback salmon, sockeye, also known as red salmon, rainbow trout, also known as steelhead, or even cutthroat trout. Juvenile salmonids are the most prone to infection and death from C. shasta. The parasitized fish often appear darkened and have swollen abdomens due to the accumulation of fluid in the peritoneal cavity. Protrusion of the eyes, also known as pop eyes, mostly seen in juveniles. Once the digestive system is infected with actinospores, the disease is usually fatal due to the spread to other visceral organs. It appears that some species are more resistant to infection compared to others, and that susceptibility appears to vary by strain of salmonid. Research indicates that the most at risk are juvenile king salmon, silver salmon migrating to infectious regions, and rainbow trout. While this is of good news to other salmon species, they too are prone to infection and overwhelmed by a large number of C. shasta parasites. Containing some of southern Oregon and much of northern California, the Klamath River Basin is a region of interconnected waterways with alarmingly high parasite densities. Much of Charlene's research has taken place in these streams and rivers that make up the Klamath River Basin. The Klamath River is of concern due to its waters being an ideal environment for C. shasta. This is in part due to the increased water temperatures, high pH values, and low dissolved oxygen concentrations. Many regions in the river house dense beds of cladophora, which is a filamentous green algae. This algae serves as a secondary habitat for Maniukia speciosa, resulting in increased worm abundance. This allows for increased myxospore infections to the polychaetes, resulting in higher occurrence of ceratomyxosis. What appears appears to be the most important environmental condition needed, besides fresh water, are warmer temperatures. Incidence rates of enteronecrosis increase in the summer months when water levels drop and temperatures rise. This is of concern because we are continuing to see increases in temperatures year after year. Even though the water levels in Northern California are mostly abundant, they must pump much of this water down to the lower part of the state. In order to fix this dilemma of proportioning water, dams were built in much of the Klamath River basin. Though these dams provide much water to the dry regions of Southern California, they may be playing a role of increased sea shasta infections. Many conservationists would like to see the dams removed so that the native environment can return. They argue that the dams, like the Iron Gate Dam shown above, limit the ability for fish to reach their native regions. They also argue that removing dams would result in less stagnant water, reducing polychaete worm environments. While others believe that removing the dams would allow genotypes not present in the regions further up the river to be influenced by genotypes below the dam, this could result in higher susceptibility to strains of salmon that are not very susceptible right now. In the 2007-2008 Klamath River fishing shutdown was not completely a result of sea shasta. It is hard not to believe that the parasite affected the decision. Due to very low returning salmon in 2007, the state was forced to shut down much of the river to fishermen. It is outbreaks like these that have called attention to parasitic interactions in the salmonids. Fishing lobbyists play strong roles in Washington, resulting in increased awareness to these threats. Without these people, money could not go to parasitic research. Even though awareness of sea shasta is now prevalent in the fishing community, the parasite continues to devastate particular regions, most notably the Cowlitz Fish Hatchery in Washington, where year after year outbreaks kill thousands of Chinook salmon on a yearly basis. What can we do to stop further infections? There is no current vaccine. Much research is being done on sea shasta. Both Oregon State University and Humboldt State University have research teams dedicated to further understandings of this unique parasite. Along with these universities, many organizations also play roles in limiting sea shasta infections, such as the U.S. Fish and Wildlife, Bureau of Reclamation, the Karuk Indian Tribe, the Yurok Indian Tribe, and the National Oceanic and Atmospheric Administration. Though more money and research are being deployed, it is very hard to conduct field research on sea shasta. Many fish often die and are eaten before prevalence statistics can be noted. The fact that salmonids travel from freshwater to marine water back to freshwater makes it extremely difficult to repeat conditions in the lab. If climate change continues to persist, we could expect to see more sea shasta infections in Oregon, Washington, British Columbia, and even Alaska. But in the end, I feel that sea shasta is a very curious and unique parasite. I am proud to say OSU is one of the lead research teams regarding this parasite. I hope you all enjoyed my presentation of parasitic interactions